Good day, everyone. We are the Group 5, and in this report, we are going to be reporting our critical review about our activity on the suppression of autophagy and osteocytes mimics skeletal aging, which for the introduction, Ms. Season will be tackling. For analysis, Mr. Mamwad and Ms. Agregado, that will be concluded by yours truly, Ms. Pasco. So, let's delve into it.
Bone health is essential for overall physical well-being as bones provide support, protect vital organs, and enable movement. Throughout our lives, bones constantly remodel themselves, balancing the process of bone formation by osteoblasts with bone resorption by osteoclasts. However, as we age, this balance can become disrupted, leading to a decrease in bone mass and an increased risk of fractures and conditions like osteoporosis. Recent research has started to reveal the complex biological mechanisms behind this process, particularly focusing on autophagy, which is a critical intercellular recycling mechanism that helps maintain cell health by removing damaged components. The study emphasizes the important role of autophagy and osteocytes, the most common bone cells that play a key role in regulating bone remodeling. By exploring how the suppression of autophagy in these cells mimics the natural aging process, this research offers valuable insights into the physiological changes that occur in bones as they age. It highlights the complex relationship between autophagy, oxidative stress, and bone health, suggesting that a decline in autophagy may significantly contribute to age-related bone loss and fragility. As we examine the physiological aspects of bone formation and aging, this review will assess the findings of the study and explore the implications of autophagy on bone remodeling. Additionally, it will consider how these insights could guide future treatments aimed at preventing and addressing osteoporosis and other skeletal disorders in older populations. Understanding the connection between autophagy and bone health is vital for developing effective strategies to maintain bone integrity and enhance the quality of life for older adults. By focusing on these relationships, we can better appreciate the broader significance of autophagy in the aging process and its potential as a target for intervention in age-related skeletal diseases.
Autophagy serves two key functions. It provides energy by breaking down cellular components during nutrient shortages and recycle damaged proteins and organelles. When autophagy is suppressed, it leads to an accumulation of defective mitochondria, increasing reactive oxygen species production. This recycling process is vital for the health of long-lived cells and decreased autophagy efficiency and is linked to aging. Genetic suppression of autophagy in neurons and myocytes show aging-like effects, highlighting its role in cellular health. Autophagy plays an important role in the terminal differentiation of some cell types. For instance, mice that lack autophagy in their red blood cells precursor develop serious anemia and usually die for 14 weeks. To see if autophagy affects how osteocytes develop from osteoblasts, researchers checked the density of osteocytes in the certain bone areas and found it unchanged in the mice that had autophagy suppressed. The rate of dying osteocytes was similar between the two groups, and electron microscopy showed no significant differences in the structure of newly formed and mature osteocytes. Additionally, levels of specific genes related to osteocytes like SOS and MEFE were also unaffected in the altered mice. Mayatsan box?
Osteocytes in autophagy would maintain bone health through bone turnover and structural strength modulation. The cells are raised from osteoblasts that sense mechanical stress and are responsible for bone formation by osteoblasts, as well as bone respiration by osteoclasts. Studies where mice had an autophagy suppressed because of the deletion of their ATG7 gene revealed major decrease in bone mass, particularly in their cortical and cancellous bones, which resulted in higher porosity and weaker bones. This might shows delays in their bone remodeling activities with fewer numbers of both osteoclasts and osteoblasts, which resulted in slower bone turnover. This is one of the characteristics typical of aging bones. Although differentiation and apoptosis of the osteocytes were not affected, Increased levels of oxidative stress, mitochondrial content, and other signs of cellular injury imply that a failure to execute autophagy accelerates bone aging. It is interesting to note that without an increase in bone mass, expression of the key regulators of the activity of osteoclast, RUNC-L, and OPG was not altered. A great emphasis of the study would indicate that autophagy regulates bone turnover. Suppression of autophagy leads to a profile similar to aging, which is bone loss and fragility. This would bring this cellular pathway into a perspective as possibly being a medical focus in diseases like osteoporosis, where there is inappropriate bone mass with abnormal turnover.
In conclusion, the study of autophagy in osteocytes reveals its critical role in maintaining bone health and structural integrity, especially as we age. Suppression of autophagy in these cells results in skeletal changes that mirror aging, including reduced bone mass, increased cortical porosity, and an imbalance between bone formation and resorption. These findings suggest that autophagy plays a key role in preserving bone homeostasis by mitigating oxidative stress and maintaining cellular health. As such, targeting autophagy pathways offers a promising therapeutic approach for preventing age-related bone disorders like osteoporosis, potentially preserving bone mass and strength in older populations. And that's it for our report. We hope you learned something and we hope you enjoyed. Thank you.
Nice one.